In the last session, I talked about how the finished human genome sequence was prepared, beginning with the initial draft sequences, and all the work and effort that went into getting very large, accurate sequence segments. And these proved to be extremely valuable for the next phases of the project, which were really revolutionized by the development of next generation sequencing applications, and the critical complementarity of the finished genome sequence and the next gen applications is perhaps underappreciated, in that because the next gen sequencing applications generated by and large short sequence fragments and did not provide a simple way to get to whole genome assemblies. Almost all of the next-gen sequencing applications are directly dependent on the mapping of next-gen reads to the human reference sequence. So the high-quality reference sequence of the human genome turned out to be critically important for these next phases of applying genomics to human biology. I also spoke about the initiation of the next-gen sequencing methods, a little bit about the methodology. Today I'm going to talk about a few specific applications, and also talk about a couple of the large-scale genomics projects that have been launched that have really leveraged the next-gen sequencing applications. These are meant to be a sampling. They're certainly not comprehensive, and other later lectures in this course we'll talk more specifically about some of the additional next-gen sequencing applications, particularly ChIP-seq and RNA-seq, which I won't discuss here. So next-gen sequencing methods really drove down the cost of sequencing to the extent where you can now actually get a whole genome sequence for less than $10,000. Some people say 7, some people say 5, with the ultimate goal being able to get a full human genome resequencing done for about $1,000 or less. And once you reach that level, it becomes a tool that's very affordable and can be incorporated into routine medical applications. The initial impact of the cheap DNA sequencing is that a whole plethora of applications were developed to probe biology. Whole genome resequencing we've talked about, exome sequencing is very similar to that, but also sequencing all of the RNA in a genome, sequencing all the microRNAs, doing experiments where you immunoprecipitate proteins that are bound to DNA and finding out where all those localization sites are in the genome, ChIP-seq. DNase I hypersensitivity, doing experiments where you probe the binding of proteins to the RNA sequences. All kinds of different innovative and imaginative applications were developed based on the ability to sequence lots of DNA very cheaply and generate sequence tags that can be mapped back to the human reference sequence. The large-scale genomics projects, of course, the original large-scale genomics project was the Human Genome Project. A whole series of additional projects have been developed based on the human reference sequence, including the Encode project and the 1,000 Genome project, Cancer Genome project. Those first three I'll talk about today. But in addition to those, the microbiome, which will have its own lecture in this series, as well as a whole host of other projects focusing on creating large data sets that can be shared by a particular biological community who's interested in all of the data for developing experimental systems, different kinds of approaches to studying problems and so forth. So what we'll talk about today as far as next-gen sequencing applications are the whole genome resequencing. I'll show an example of a paper that uses this. Exome sequencing, similarly, I'll briefly introduce the method and then go through a paper that uses this, as well as the personalized copy number and segmental duplication maps that can be constructed from whole genome resequencing data. So whole genome resequencing, one paper that I selected that's an interesting case where this was applied is where 10 genomes from hemophilia patients and 10 control genomes were all sequenced to high depth of coverage using paired-end Illumina sequencing. So you had 75 base pairs, paired-end sequencing done on these genomes. And I already mentioned that like all these next-gen methods, the data requires, as an initial step, the alignment to a reference sequence. And through those alignments and comparison with the reference sequence, you can detect all of the single nucleotide variation. You can detect all the point mutations, all the single base changes between the genomes. And very similarly, small indels can be detected and characterized using the reads themselves. You need to use the paired-end information to get information on structural variation in larger deletions, insertions, inversions, translocations, and so on and so forth. So paired-end reads are actually quite important for the success of this approach. A reminder once again of the pipeline for the Illumina sequencing. You go from the massively parallel sequencing to base calling to the alignment of the high depth of coverage reads to the reference genome. And by comparing the aligned reads to the reference genome, you can detect the variations, the small variations directly from the read alignments, and the larger variations, the structural variations and so forth, by looking at the paired-end organization of the reads from the fragments in the library. This table summarizes the data and the depth of coverage in this particular data set, and you can see that there's between 20 and typically 50x coverage of these genomes, and very, very good coverage of all the exomes. And this is a whole genome data set, so the entire sample was converted into paired-end format and sequenced, and then mapped back to the reference genome. One thing to remember is that not all of the data is mappable back to the reference genome, particularly in cases where there's a lot of repeat structure and segmental duplication content. The reads cannot necessarily be uniquely mapped back to their source in the genome. So there's always going to be holes in the analysis of these whole genome and exome mapping experiments, exome sequencing experiments. So an interesting characteristics of these data that was seen early on is that while you do see all of the variation that's been seen before in these data sets, there's always a set of new information with each genome. And these were unrelated individuals, so they shouldn't necessarily share large haplotypes or anything between them. And that is that there's about 12 to 15% of the SNPs and the small variations that you see are always novel. And this suggests that there's a significant fraction of human variation that's either private or very rare in the population. And this is perhaps interesting, and it's certainly interesting, perhaps quite important in thinking about human variation and the kinds of variation that can cause phenotypes. A lot of the initial studies using HapMap were based on common variants and trying to do association studies of those haplotypes that contain the common variants with different human phenotypes. It turns out that a very significant fraction of the variation in the human genome is caused by very rare variants or even private variants. And so the ability to completely sequence the genome is going to be really critical in trying to understand human variation and human disease. This slide shows the concordance between the genotyped SNPs and for all these genomes, they carried out conventional genotyping analysis for the common SNPs, and when they compared that with the SNPs that were seen using the whole genome sequencing analysis, they found typically, and this is also typical of all these experiments, they find a very high concordance, but not a perfect concordance. And many of the non-concordant reads turn out to be sequences where the SNP that's being interrogated in the genotyping experiment has a second SNP very close to it, and that second SNP prevents the proper annealing of the oligonucleotides that are required for conventional genotyping. And so you can only detect these kinds of variants using the sequence-based approaches. Also, a large and perhaps surprising number of coding region mutations were found in all of these genomes. Interestingly, when there were indels in the coding region, they were enriched in every third nucleotide, and this is consistent with a preferential insertion or deletion of nucleotides that correspond to a complete amino acid, so that you don't frame shift your protein with these mutations. Nonetheless, there were also obviously a fair number of mutations that disrupted the coding sequence, and so in every individual there are actually a surprising number of protein encoding genes that are non-functional, and not simply in the heterozygous state, but in the homozygous state. So every one of us is walking around with basically null alleles of some protein encoding genes. Now, I mentioned earlier the patients that were sequenced, whose DNA were sequenced here, were selected. 10 of them had hemophilia A, and the known major mutation for hemophilia A is known to be in the F8 gene, and this was confirmed by the whole genome sequencing experiment for 5 of the 10 individuals. And one thing to note here is that other genes that were found to be mutated and deleted homozygously in individuals were also found to be homozygously deleted in the controls, and that's not the case for the F8 gene. And so it's important to always have the ability to compare the genomes of the affected individuals with control genomes, so that you can get a sense for which genes are preferentially affected in the disease individuals, because there are always going to be a fraction of genes which appear to be mutated and which in fact are mutated, but may not have any functional consequence. So in looking at all of the data from these genomes, I already mentioned that there's a certain level of private or rare variance in all of the individuals, and that number, at least in this data set, comes to about 140,000 or so single nucleotide variations that are very rare or even private, and that seems to be a level that is likely to be present throughout most of the population. In addition, the way that they determined the homozygous null mutations in these genomes, which was a little bit more loose than other studies did, they found an average of 165 homozygous null mutations, which means that genes that are predicted to encode proteins are deleted or non-functional in both alleles in the particular genome. So out of the 20 to 25,000 protein encoding genes, there's a small number but significant number in any given genome that appear to be non-functional. So whole genome sequencing is obviously extremely important and powerful technique, but a more efficient way to get at the protein encoding fraction of the human genome is to use a targeted sequencing approach. Other applications of targeted sequencing include selecting particular chromosome regions that may have been identified through genome-wide association studies or other kinds of studies, and targeting those chromosome regions for resequencing in a large number of individuals, perhaps a number of disease cases compared to a number of controls, to try to determine which actual sequence variants may be causing the phenotype that you're interested in or the disease that you're interested in. So targeted sequence capture followed by massively parallel sequencing has evolved as a very powerful technique and a way to very cheaply, relatively speaking, get at the DNA sequences you're the most interested in from a very large number of samples. And the way this method works, and I'm showing it in this case for a region that I'm interested in, the human subtelomere region, is that you start out with genomic DNA and you create a library of fragments and put on the Illumina adapters as one typically would. In my case, I'm actually using a sequence tag to physically tag the tips of the chromosomes. Once you have the fragment library, you go through a targeted sequence capture step, and I'll show you the two main ways that that's done in a moment. But once you've selected the DNA from the whole genome using this targeted selection method, you amplify that pot of DNA and go through the high throughput paired end sequencing. The targeted sequence capture, the two main ways to do that are, first of all, through a custom microarray, in my case a subtelomeric oligonucleotide microarray that is capable of annealing to the complementary DNA that's in the Illumina library that's been prepared, and it sticks to the array, and then you wash away the non-specific DNA and elute the specifically binding DNA, and you have your selected target region. A similar approach, but using RNA instead of DNA, is to start out once again with a large set of long DNA molecules, but create RNA replicates of those that are biotinylated. And the RNA that's biotinylated is then used to do a solution hybridization reaction with the Illumina library. And once again, the complementary DNA sticks to the RNA, and you can use the biotin moiety to effectively pull out, affinity purify the annealed parts of the genomic library, and then you can digest away the RNA and you're left with the DNA that's been selected. So either of these two methods actually works extremely well. If you make your targeted microarray or your long RNA pool specific for oligonucleotides that cover only coding regions of the human genome or the exons, it's called an exome microarray. And there are now commercial exome microarray approaches that are available that very efficiently enrich the Illumina libraries prepared from whole genomes for only those fractions of the genome that contain the exons. And these provide a very powerful approach for very efficiently getting at the protein coding regions of the human genome. This paper out of Lifton's lab shows an extremely powerful approach of exome capture. And here they're looking at individuals, and in particular one individual who has a phenotype that looks like something called Bartter syndrome, which is a renal disease, a kidney disease. It turns out that after doing exome sequencing on this individual, they actually identified the mutation as being in a chloride channel, and so the individual actually did not have classical Bartter syndrome, but instead a condition called congenital chloride diarrhea. And this is just one example out of literally hundreds of examples that are now available where exome sequencing has been done on individuals with particular diseases, and the causative mutations have been found. So to take a quick look at the kind of data that you get from these exome capture experiments, in this case there were 5 individuals who were sequenced, one of whom had a phenotype that was of interest in that individual, so the GIT 284 individual. And you see you get very similar kinds of statistics from normal individuals and disease individuals in terms of the numbers of missense mutations that are found and the number of premature termination mutations that are found, splice variants, and so forth. So there are always more of these kinds of mutations that are expected to lead to non-functional proteins than can actually cause the disease. So a critical step is always that last step to sift through these candidates and try to find the one mutation that's actually believed to cause the disease. And the way that that's typically done is to take advantage of other kinds of biological information that you might have. For example, in this case, they looked at, of all of those mutations that caused what appeared to be non-functional proteins, which of those proteins were actually highly conserved evolutionarily? Where did the mutations occur? Did they occur in an evolutionarily conserved part of the protein where the change in amino acid might cause a mutation that would cause a non-functional protein? And they narrow their list of candidates using that kind of an approach. And by doing so, they come up with a single or just a few that they can then test individually using PCR-based approaches on additional patients that have similar kinds of conditions. So this describes the sensitivity and the coverage of the exome sequencing itself. What one can see from panel A is that while you get a high depth of coverage for most of the bases represented in the exons, there is always a small subset that coverage is rather poor. And this corresponds to what I mentioned earlier in terms of very GC-rich and very AT-rich segments of the genome. Some of these occur in the exonic regions, and coverage is not complete. So it's one of the drawbacks of the approach, but it's a fairly small fraction, and you can actually capture greater than 93, 95, 97% of the exome sequence with 10 or more reads covering each base, which gives you almost complete coverage, but not totally complete coverage. B shows the error rate of these short reads. As you get longer and longer reads, you increase the percentage of error. Even as you approach 75, though, you're still less than 3% error per base. But clearly, one needs to take that into account when looking at potential, what you identify as potential mutations. You want to make sure that you have sufficient read coverage and that different parts of the reads cover the candidate mutation, so that you can be sure that the difference that you're seeing is actually real and not an error. Coverage in both cases, the depth of coverage reaches a plateau at about between 20 and 50x typically. So by the time you get to 30 or 40x coverage, you're getting all of the information that you're likely to get from these experiments. Now, in this particular paper, the affected individual is actually part of a large family that included some consanguineous marriages, which means first or second degree relatives were marrying each other. And in those cases, you would expect much of the genome to be homozygous, since the related individuals were mating with each other. And because because of this family structure, one would expect to see a recessive gene that would be homozygously deleted in the case of the mutation. And that's in fact what they found in this particular study. They found a single gene that had a homozygous change that it was predicted to create a non-functional protein in this particular chloride channel. Now, importantly, this was still a candidate mutation in that it was only found in this one individual amongst other possible mutations that might have caused the disease. But when they went into a pool of additional patients with a very similar kind of phenotype, they found that in fact in this very same gene, they found many additional mutations that were capable of causing the same phenotype, which is basically this chloride deficiency, and in each case the mutations caused a non-functional protein. So a third application I wanted to talk about very briefly is how you can actually use the whole genome data sets to look at DNA copy number and copy number variation in individual genomes. Now, we talked a lot about SNPs and SNP variation in small nucleotides, and we talked to some extent about structural variation, including insertions and deletions and inversions. Copy number variation is a kind of structural variation where you have greater or fewer numbers of copies of a given segment of DNA. And the genes that are embedded in these copy number variants can obviously be present in more or fewer copies depending on what the level of how many copies of that particular variant an individual has. So the way that the whole genome data sets are used for this is to simply count the number of reads all the way across the genome, in this case in 100 base windows. As you slide those across the chromosome, you do a count of how many mapped reads there are, and in cases where you have fewer numbers of copies in a source genome relative to the reference genome, you get what appears to be a deletion, and where you have more copies, you get what appears to be an insertion. And this simple principle was applied genome-wide, and algorithms were developed to very efficiently enable detection of these copy number variants. This shows a particular region of chromosome 1 where the data indicates a fewer number of copies relative to the reference sequence. And this slide shows several different regions of chromosome 1 in different individuals, where you can see that different individuals have different numbers of copies of those particular segments in each of the different cases. And these are from different segments of chromosome 1. So clearly, DNA within these segments are in different copies in different individuals, and any genic content, any genes that are present, would be present in more numbers, in higher numbers in people with duplications and additional copies, and in fewer numbers in people with deletions. So the statistics from this particular study along chromosome 1 are shown here. You can take a look at that. Now, in a more thorough and broader study that was done by Alkin et al. In 2009, a genome-wide assessment was done of this copy number variation, and the list of the 30 genes in the human genome with the most variable copy number are shown here. And this is quite interesting because you can see the very broad differences, the very dramatic differences in many of these genes, all of which have functions in the human genome. And one of the challenges is to be able to detect these variants and ask the question, what does it mean to have a much fewer number of some of these genes in an individual, or a much greater number of these genes in an individual, and that's still an open question. There's there are a number of studies that suggest that copy number variant regions perhaps play a role in certain diseases, and there are follow-up studies underway to determine whether in fact that's true. So the large-scale genomics projects are once again based on the human reference sequence, and they have a number of properties in common with each other. They're almost always interdisciplinary and involve large consortia, which are large sets, large numbers of individual laboratories contribute to them. Computational bioinformatics resources are critical for all of these projects to be able to handle the huge amounts of information that are being produced, to organize the information, and to figure out ways to display the information that scientists can understand and people can use to gain access to this information. Almost always there are very rapid release policies and public data access to these data sets, and there are typically large scientific communities that benefit from each of these large-scale genomics projects. So there are centralized websites, data centers that analyze the data and distribute it to the whole community. As I mentioned, the human reference sequence is the foundation for many of these large-scale genomics projects, but I should mention as well that the model organisms where the reference sequences are finished also form the foundation for a number of large-scale genomics projects that are similar in principle, but focused more on understanding the function of the model organisms and processes that can be studied in the model organisms. All of these projects are heavily dependent on Nextgen sequencing to very rapidly and cheaply generate information on all kinds of different aspects of the reference sequence. So the 1,000 Genomes project is one such large-scale genomics project. It grew out of the HapMap project, which was a project to understand the common variation in the human genome. And the 1,000 Genomes was an effort to use Nextgen sequencing to probe beyond just the common variation to identify all of the sequence variants that are present in at least 1% at a frequency of 1% in the population, but also get information on a lot of the rarer variants and create a database of this information and make it available in forms that people can use for their genetic studies. So the initial plan for the full project is to sequence about 2,500 genomes at 4x coverage, and to use that information to get SNPs, primarily SNP information. Because any particular region of a genome contains a limited number of haplotypes, and this was known from the HapMap project, data that could actually be combined across lots of samples to allow the efficient detection of most of the variants in a region. If you had all of this information from all these different genomes, you'd have multiple copies of each of the major haplotypes, and you could leverage that information. So combining the data from all these samples would allow highly accurate estimation of variants and genotypes for samples that were not seen directly, perhaps by the light sequencing of individual samples. So the way this data is being used, they're combining this 1,000 genomes data with genotyping data from GWAS studies that use primarily tagging kinds of SNPs to identify additional variants beyond the SNPs that are genotyped directly. And once again, this is based on the understanding that there are a limited number of haplotypes in the population, and so you can predict from the 1,000 genome data where some of these additional SNPs are likely to map, even if you haven't genotyped them directly. And that's a process called imputation. So The data is available on almost all of the variants with a frequency of at least 1%, plus a large number of additional rare variants. And these are used in population-based studies of recombination selection and looking at different kinds of population structures. So just to give you a sense of the HapMap, since we really haven't talked about that very much yet, this is a screenshot from the HapMap website of a particular region of the genome, and the DNA is organized into regions of linkage disequilibrium that vary from population to population. And all of this information was gained initially through genotyping experiments using common SNPs. With the 1,000 genome data, you can overlay this rich data set of additional imputed genotypes on top of the common variants to more precisely define intervals where associations between the variants and disease phenotypes occur in the genome. So the 1,000 Genomes project led directly to the concept that personal genomes may in fact be very valuable tools in terms of understanding an individual's susceptibility to disease. And personal genomes, as the term implies, is the complete sequence of an individual's genome. And so in addition to the common variants that one would be able to find using genotyping, you'd also get access to all of the rare variants and private variants. And the idea is to use this information to help define disease susceptibilities. If you know you're susceptible, for example, to heart disease, you may be able to modify your behavior to live a healthier life. In addition, if you have susceptibilities to certain drugs that could be identified by sequence variants in your genome, then you'd know which drugs to avoid, which drugs are likely to be effective for particular diseases. Importantly, for specific diseases like cancer, where actual genome mutations occur that lead directly to the disease itself, you may be able to use the particular changes that occur in the cancer genome to figure out which particular drugs, in theory, might be the most effective in treating that particular cancer. Most of the promise of personal genomes has not yet been realized. There have been individual cases and a few cases where it's actually been used very effectively. But while it's fairly straightforward now to get the information, it's still very difficult to try to associate that information with particular diseases and susceptibilities. There just isn't enough information available yet, enough biological information available to understand what the variants mean. So a second important large-scale project is the Encode project. And the Encode is basically an attempt to assign function to DNA sequence. And that's done at many levels, at the level of annotating all of the genes in the genome, looking at all of the regulatory sequences in the genome, and looking at, more recently, the epigenome and how the locations and organization and patterns of histone modifications in particular cell types under particular conditions may be affecting the function of the genome and of the cell. And so all of the next-gen sequencing methods that I talked about earlier are applied to gathering information that can then be mapped back to the reference sequence to give us clues as to what kinds of functions are happening in the DNA. All of this information is collected on a single website hosted by UCSC, and it's a very data-rich and powerful tool for comprehensively organizing all of the information that we have that's related to that particular segment of DNA. And an example of that is shown here, where we're looking at a 40 or 50 kb region of the genome, and the RNAs that are identified in the gene models at the top of the figure, and below you have tracks that you can actually select amongst all of the data that are available from the Encode project to look at things like the expression of RNA sequences in different cell types, the patterns of histone modifications, the patterns of hypersensitivity sites, DNase I hypersensitivity sites, methylation patterns under different cell types and different conditions, and the organization of DNA sequence repeats and duplications, copy number variants in a given region of the genome. And all this information can be organized and laid out in tracks underneath the genome sequence itself to try to get as comprehensive an understanding as one can of the functional elements that are present in that segment of DNA. So the final large-scale project I wanted to talk about is the Cancer Genome Project. And this is an international effort to get at the genetic causes of cancer. And of course, cancer is not a single disease. It's hundreds, thousands of individual diseases. And each cancer has its own characteristic biological properties. And in fact, each cancer can be divided into subtypes that have characteristic features that they share. And ultimately, each cancer is essentially an individual disease. The way cancer is dealt with by an individual is going to be different depending on the person. So the ultimate power of the Cancer Genome Projects is to gain enough understanding of the different types of cancer and the subtypes of cancer and the different stages of cancer so that one can identify targets for intervention and perhaps develop ideas for predicting which cancers are likely to be aggressive or non-aggressive, which cancers are likely to be susceptible to intervention by different kinds of therapy, whether it be radiation or different chemotherapeutics, or more recently, molecularly targeted therapies that are designed to disrupt particular pathways that are known to be mutated in the particular cancer type. So once again, Nextgen Sequencing has taken a lead role in this large-scale genomics project in terms of understanding the mutations that occur during tumor progression, as well as the gene expression changes that occur, and developing and understanding what pathways are affected, what genes are affected. In this particular paper, they looked at breast cancer, and they looked at the copy number changes that occurred in a large number of individual breast cancers, as well as the mutations that occurred by doing complete exome sequencing of 100 different cancers, breast cancers here. The two major categories that the cancers clustered in using estrogen receptor positive or estrogen receptor negative typing are shown on the left and the right respectively. The individual cancers are represented by each by each column in this figure, and individual genes that were found to be mutated are shown in each row in these different cancers. And one can see that there are different patterns of mutations that can be identified in the cancers, and different mutations and copy number changes. And the take-home message from this particular paper was that they found a number of additional genes that had not been discovered previously in breast cancer, and those are listed, MAP3K1 and MAP3K13, AKT2, etc. They also found several non-overlapping driver mutation patterns, and driver mutations in cancer are those mutations that actually lead to tumor evolution and cancer progression. And there are lots of mutations in cancer genomes, many of which have no functional role in the cancer process itself. So one of the major challenges is and remains understanding which of the mutations are actually driving the cancer, and which ones are just occurred during the process and are taken along for a ride, the so-called passenger mutations. So they found several non-overlapping driver mutation patterns suggesting that those groups of cancer with the different patterns are likely to have different biological properties, perhaps be more or less susceptible to different kinds of therapies, and so on and so forth. They also found in this study that there are several new genes that they found are present in the same signaling pathway, the Jun kinase signaling pathway. And this suggests that this particular pathway may be quite important for breast cancer tumor progression and highlights a particular pathway that might be targeted in developing drugs or trying to figure out how to intervene and cure the cancer, or at least inhibit the cancer. So that was just one study and one cancer genome project. All of the information from all of the mutations from all of the studies that are being done are being gathered in a centralized database called Catalog of Somatic Mutations in Cancer, or COSMIC. And this is a way to collate all of the information in a central location. And so not just the mutation information, but also information on what publications are associated with the studies, the types of samples that were used to do the sequencing, what kinds of cancers were involved, what their characteristics were, whether they were in situ or invasive tumors, whether they were recurrences, whether they were metastases or cell lines. All this information is gathered into this single database that can be queried from a number of different perspectives by the tissue type or the cancer type, or by gene to look at all the different kinds of changes that happen in particular genes. This is an example of the gene-centric query. This is the BRAF gene, and the purple areas show where mutations have been found in the gene in particular cancers. And you can see where the different breakpoints have been found to occur in this particular gene in different cancers, as well as the copy number changes, loss of heterozygosity, and amplifications. And you can drill down into each of these categories to get additional information on to help understand the role of BRAF in tumor progression in these different kinds of cancers. So in addition to the consolidated data, you could actually also access the raw data itself by accessing the Cancer Genomics Hub, which is a repository for all of these data sets. And in this case, you need to be registered for this, because this is obviously patient information. But once you validate that you're a researcher and agree to certain protections of the data, then you can actually dig into this data and do your own analysis of the data to try to understand it from whatever hypothesis or perspective that you're interested in. So really this highlights a major value of these large-scale projects, in that you're leveraging all of the data from all of the groups that are doing these analysis, so that if you come up with a new analysis technique, you can immediately go to this rich data collection and analyze all of these different kinds of data sets using your technique. In doing so, any individual laboratory has access to the data from all of this large group of consortia that generated the data initially.